Hello everyone, my name is Panlu, and today I'm going to demonstrate to you our EPID Immune Atlas platform. EPID stands for Extended Pediatric Immunome Characterization. So in this platform, we have a database which consists of 114 healthy individuals, ranging from cord blood samples to newborn individual samples and all the way to adult samples. So in this platform, scientists or researchers from all over the world are able to upload their which is their mass cytometry data, onto our platform and run through our pipeline analysis automatically and get to compare it against our database. So before any comparison is to be made, let us look at our database first before we start to explore the analysis process. Users could click here to view the healthy database. The web page will direct users onto this page. It will show you the basic information such as the phenodata of the database. So here are the cell counts of every age group in our database. And there are some distance plots such as the MDS plots as well as the PCA plots for all our samples in our database. And besides that, it will also show you the distance between the markers in our database as well as what are the markers that are most important in our database. So this is to aid users who wants to do some prediction models or to do some analysis on the clusters by only choosing the most important markers. So NRS score, which stands for non-redundancy score, those markers with high NRS score are those markers which are important to this data group. So let's say when I look at adult samples, and these are the few markers which are important. And if I look at cord blood, the plot will change. So this allows some scientists or researchers to look into our database very carefully. And also here shows the t-SNE plot of all our 114 samples, and users could click on different age group to view. And these are the media expressions of the markers, so users could click relevant data information and look at it separately. And also, this is how we choose the clustering. We also shows a diagram, which is the PAC plot, which stands for the proportion of ambiguous clusters. So as K, which stands for the number of clusters, increases, so proportion of ambiguous clusters will decrease until it reaches a plateau. So in our database, we chose like 80 nodes here because the method we use is called flowsum. So flowsum will cluster all nodes into its SOM codes. SOM codes behaves in the t-SNE dimension, and this tells the users, say we chose K80, will K80 be a perfect cluster, or there will be some overclustering. Plot over here in t-SNE dimensions or PCA dimensions will show you whether is there any overclustering being observed. So each color represents one node, and if those circles of the same color are grouped together and they are far apart from other colors, which means that our cluster is perfect. So here shows marker expression of each node, so users can determine the phenotypes of each samples. And here shows our database of 140 samples. So this shows the volcano plot, and here shows that relative abundance plot. These nodes are grouped according to their node frequencies. So users could observe what are the patterns and the phenotypes as well. So phenotypes will be shown here, as you could see from the screen. These are being automatically generated from our pipeline. Users could also compare this result with the previous heat map shown in the previous page. These are the SOM codes, and they are grouped according to their cell types. So each color now represents the cell types. And here shows the t-SNE plot of our 114 samples, and each color represents the cell type. Users could look into our database in this way as well. There are some reclustering being done because according to the some t-SNE plot and PCA plot is shown, there are some nodes which are over clustered. So we regroup them according to their cell types because we realize some of these nodes belong to the same cell type. So we regroup them, do the reclustering, and show in different colors as shown here. Okay, here shows also the relative abundance, but instead of the nodes, it shows the cell types. And also, after that, 3D volcano plot again will be shown on our database. Instead of nodes, here shows the cell types. So you could observe what are the cell types which are high in which group. So these are our database results. And also here shows the cell proportion changes across the age group. So from the left, cord blood newborn, all the way to adult. And each color shows specific cell type. So you could see the cell type proportion changes across different age groups. And lastly, here shows the proportions of each cell types across age groups in a more separated way. For example, the B cells starts to increase when the age increases. So this gives a clear background and knowledge about our database. So now imagine I am a scientist X, and now I'm going to upload my data and compare with the current database. So I'll do some uploading of the data, which I have already done, and there'll be some loading process. The analysis pipeline on our platform will be carried out for my data, and it will be compared against the current database. So here shows the result. So I'm using HPV samples. As you could see from here, these are the phenotype differences in terms of the cell count. And also here will show me the distance plot between my samples. And here are the database results. When my samples are added to the database, and the marker's distance will change as well. And the importance of each marker will change according to this plot, as well as the NRS plot. And also the t-SNE plot of the samples will be plotted, and each color shows each age group, and here shows my result and here shows the database results. And also the media expression of the markers and the recommended K according to our choosing criteria. For my case, it will be 87 nodes. So I will look into here and then see whether is there any overclustering. Say for here, there's some overclustering of the nodes because different colors, which represents different nodes, are clustered together, even though they are separated as different nodes. So I'll look into their phenotypes separately using this heat map. So to aid our t-SNE dimension, we also look at the PCA dimension. And after I look into the marker expressions, I will upload a data sheet like this. So there will be two columns. One shows the node ID, the other shows the cell phenotype. And it will automatically generate for me the results here. It will show me how my HPV samples in each node are compared against the other age groups in our database, and also the relative abundance in each group when my samples are combined with the healthy database. The colors will be reassigned according to cell types in the SOM t-SNE dimensions as well as the PCA dimensions, such that I will look at them separately. When you move your mouse over, you will also show the cell types that you upload in your datasheet previously. So the color are determined by the datasheet you uploaded. The t-SNE plot of the cell types will also be shown. So these are my HPV samples, and this is the database. As I only uploaded 2 patients with HPV, so I will select their age group. They are all of age 14, so I will look at age 14. So I will look at how the cell types changes across different cell types, as well as just in age 14 samples. So for instance, I look at the memory cells and the CD4 cells. And also, the pipeline will do automated reclustering after some merging according to the cell types you have identified. And also, it will show the relative abundance again, but in terms of the cell types. And the lineage marker expressions as well as the functional marker expressions. One good thing about the functional marker expression is that, let's say when I click on the adults, it will automatically arrange the marker expressions according to their values. And these are the markers with low expressions, and these are the markers with high expressions. So this will tell you further information about their marker details in specific expressions. After this, I'll look into the 3D volcano plot and it will tell me what the cell types being observed in my age group. So for instance, I could see that in my age 14 HBV samples, the naive T cells are very high and double negative T cells as well, and also the B cells. And I can do a direct comparisons with the database here. And also, the cell proportions as well will be shown here, and I will look into my own age group over here, which is the HPV samples here. So I can compare directly, not just in terms of the age group cell compare against my age 14 samples over here. I could also compare across all the other healthy samples against my HPV samples. And lastly, this shows a similar information as compared to above. And if I want to look at my HPV samples, I could see very clearly that each cell type proportion difference in my samples as compared to the healthy samples in age 14 as well as across all other age groups. You could see that my naive CD8 T cells is much lesser than those ones over here, and also the other cell types. This is the end of the presentation. Thank you for listening. I hope you have a better idea of what our analysis panel is about. Goodbye.